Good day, my lecturer, my viewers. I am Okechukwu Jesus Mercy, a student of Molecular Genetics and Biotechnology in University of Nigeria, Nsukka. I'll be talking on the topic description of cell signaling pathway leading to apoptosis. But before I continue, I'd like us to know what apoptosis means. Apoptosis simply means a programmed cell death. Whenever a cell undergoes stress or DNA damage, it kills itself to minimize and maintain the normal number of cells in the body to avoid accumulation of that damaged cells, which would lead to cancer. There are two major pathways that initiate or lead to apoptosis in the mammalian system. They are an extrinsic pathway and an intrinsic pathway. Now, I'd like to show us the diagram of extrinsic pathway and more of how it goes. Extrinsic pathway, just like the name implies, the signal comes from the outside of the cell. So when it comes from the outside of the cell, it binds with the death receptor. When it binds to the death receptor, it initiates recruitment of adaptor protein that has death domain. They interact with each other and then they form this, which is death-induced signaling cascade. When they form that, this undergoes autocatalyzation to produce caspase 8, and then caspase 8 phosphorylates caspase 3. This caspase 3 does a very important thing. It degrades nucleus enzyme inhibitor, something that hinders the nucleus enzyme from acting. It degrades it and breaks it down, removes it, and makes the nucleus enzyme to be active. When it becomes active, it goes into the nucleus, breaks down the DNA and the chromosome, and then finally it leads to apoptosis. I'll be talking on the intrinsic pathway, which is the next one. Now, this intrinsic pathway, just like the name implies, it takes place, the signal comes from inside of the cell. The signal comes from the inside of the cell. Whenever there is an oxidative stress or DNA damage or injury, the ATM or CHK, which is the sensory protein, sends signal to P53. This P53 produces multiple proteins, as you can see in the screen. Time will not permit me to mention. And then the BAX creates a pore inside the mitochondria. When it creates pore inside the mitochondria, cytochrome C comes out. Then the cytochrome C pairs with APAF1 to phosphorylate caspase 9. The caspase 9 phosphorylates caspase 3. Phosphorylation simply means activation. It activates caspase 3, which degrades the nucleus enzyme inhibitor. And now the nucleus enzyme goes into the nucleus again, does the same thing, breaks down the DNA and the chromosomes, and finally, trust me, the cell cannot survive. It leads to what? Cell death. Thank you for watching. Stay tuned.